Hello, I welcome you all to this session where we are going to study the inhibition of enzyme action. So, inhibition of enzyme action can be of two types. One is reversible inhibition, and the second one is otherwise called as irreversible inhibition. Reversible inhibition is the temporary inhibition that can be overcome by the withdrawal of inhibition. If you remove the substance which is inhibiting it, then it will come back to its original form. It occurs due to the blocking of the active site or bonding to linkages which are required for the maintenance of active sites. So, it can be reversible equation can be overcome. Then secondly, it happens because the active site is blocked. Thirdly, it is characterized by rapid dissociation of enzyme inhibitor complex and dilution or dialysis of the enzyme eliminates the effect of reversible inhibition. Then irreversible inhibition, obviously the name indicates that it is not reversible. So it is also called as permanent inhibition. And this is due to the change or destruction of the conformation of the enzyme. And it happens because covalent bonding of heavy metals or other inhibitors of enzymes, as I have told you, like cyanide, azide, etc., presence of that results in inactivation of the enzyme. Now, irreversible or permanent inhibition can be further categorized into competitive and non-competitive types. There are some reversible inhibitors which resemble the normal substrate molecule and they compete for the admission into the active site. This is called as the competitive inhibition. And the inhibitor has some characteristics. It closely resembles the real substrate. So it is called as a substrate analog. This inhibitor will compete with the substrate and it will bind to the active site of the enzyme, and the enzyme will not be available for the actual substrate to bind. Competitive inhibition is usually reversible, and most common example is the enzyme of succinate dehydrogenase, whose original substrate is succinic acid, but malonate, oxaloacetate, they closely resemble succinate or succinic acid in their structure. They act as the inhibitors. Suppose this is the enzyme and this is the active site of it. Obviously the substrate which will bind to it will have this kind of shape. Now let us say that the enzyme remains the same, but now the substrate resembles this. So now this is the original substrate, but this is the substrate analog, not the real substrate. So once, suppose this active site of the enzyme that remains occupied with this inhibitor. So this inhibitor will never change to form the product as this and this, because it will, once this breaks up, it will form a globular thing and a triangular thing. But this squarish and triangular thing can be formed only when this substrate is bound to it. So this one is called as a substrate analog. So if this enzyme is succinate dehydrogenase, then this one which I have shaded, this is the true substrate or succinate. But this is suppose oxaloacetate. If oxaloacetate will be occupying the active site of succinate dehydrogenase, it can never form these products. So this substrate is actually not doing itself anything, not allowing the actual substrate to do anything. So that is why it is called as competitive inhibition. So when you plot it graphically, the rate of the enzyme action will be like this when no substrate inhibitor is present. But if competitive inhibition happens, the graph will become a straight line. Then let us come to the non-competitive inhibition. In non-competitive inhibition, the enzyme can have more than one active site. Suppose this enzyme has an active site like this and another active site which is present like this. This is active site 2 and this is active site 1. So once a particular, you can say, substrate binds to this second active site, suppose to this second active site, this particular substrate, it is binding. So once this substrate binds to the second active site, it will bring about a conformational change in the actual active site. Now this actual active site will become like this, and already in the second active site you will find that the inhibitor is sitting. So the original substrate for this enzyme is a squarish shaped like thing like this. So now when the active site is changed into this shape, this particular green colored substrate will no longer be able to bind to it. It is because the substrate analog is now binding to the second active site, changing the conformational configuration of actual active site, and the actual substrate is not able to bind to it. So inhibitor binds at a site which we can say is other than the active site, actual active site of the enzyme on the surface. This binding will impair the enzyme function. The inhibitor has no structural resemblance with the substrate because inhibitor is rounded, this one is squarish. It doesn't interfere with the enzyme substrate binding, but the catalysis is prevented most probably due to distortion of the enzyme conformation. Non-competitive inhibition is usually irreversible because it cannot be overcome if you increase the substrate concentration. You go on increasing the number of these square molecules, but until and unless you are kicking this rounded molecule off, it will not be having any overall effect. The most common example is the inhibition of the respiratory enzyme cytochrome oxidase. And this enzyme, if it is cytochrome oxidase, then cyanide which combines with its metallic ion has no similarity with the actual substrate which is cytochrome C. So this is cytochrome C, this is cytochrome C oxidase, and this one is cyanide. So once cyanide is sitting over here, cytochrome will not be able to bind to this and there will be no formation of the substrate into the end product. Allosteric, next we will consider the allosteric inhibition of enzymes. Allosteric regulation is the term used to describe any case in which a protein's function at a site is affected by binding of a regulatory molecule to a separate site. It results in either inhibition or stimulation of the enzyme activity, and it can be explained by the following, you can say, lines. So here, suppose the active site on the enzyme, it was looking like this originally, and here was a place where this allosteric inhibitor can bind. So once to this active site, the original substrate is not yet bound, but the allosteric inhibitor is now sitting over here. Now this allosteric inhibitor will distort it. Here it was totally changing, but it will now distort it. So now this becomes, suppose, like this. The active site is present, but it is distorted. So the original substrate, which was actually appearing like this, now this will no longer be able to hold on to this site. So that is why it is called as allosteric inhibition, because it allosterically changes the conformation of the active site. Another kind of allosteric inhibition, a substrate molecule can bind to one active site and the sub-enzyme, if it is a multi-unit system, then all the subunits, they will change their shape, and this can happen by induced fit. Suppose there are 5 active sites on an enzyme. This particular inhibitor is sitting on one site. The other 4 sites are changing their shape. Now the substrate will no longer be able to bind to it and it will be inhibited. And it is known as cooperativity. The mechanism amplifies the enzyme's response to the substrate. So here if I draw it diagrammatically, suppose there were 5 such units and on every unit there was an active site which I am drawing like this. We are talking about cooperative allosteric inhibition. So now to one of these active sites you get a particular inhibitor attached to this first one. Now the active sites which are present on 2 will change the shape like this. On 3 it will change the shape like this. On 4 it will change. On 5 it will change. So to a active site which is now shaped like this, you cannot make a substrate like this fit. So that will inhibit the formation of the enzyme substrate complex. Now let us come to the last part of our discussion, that is called as feedback inhibition. When we were studying the chemical control and coordination, this we have studied many times. Feedback inhibition is also a form of allosteric inhibition. In feedback inhibition, a metabolic pathway is halted or it is broken by the inhibitory binding of its end product to an enzyme that acts in the pathway some step behind. This step of inhibition thus prevents the cell from making more products than it is necessary, and it prevents wasting chemical resources. So now let us understand how it happens. Suppose there is an enzyme. So this is the enzyme. This will change into intermediate A in presence of enzyme 2. Suppose this step is catalyzed by enzyme 1. Intermediate A in presence of intermediate enzyme 2 will change to intermediate B. Intermediate B in presence of enzyme 3 will change to intermediate C. Intermediate C in presence of enzyme 4 will change into intermediate D, and intermediate D in presence of enzyme 5 will change into the end product. So once more and more end product is being formed, means this end product is being formed more, it will now undergo feedback inhibition and will stop the production of this. So that is feedback inhibition. It is because the concentration of the end product is increasing. Suppose a cell requires a particular amount of isoleucine amino acid. So isoleucine amino acid is synthesized from the amino acid threonine. So threonine will be attached to the active site of this particular enzyme. This enzyme, most probably it is threonine deaminase. So once threonine is attached to threonine deaminase, it will form intermediate A. Sorry, now it will change to intermediate B, C, D, and finally it will produce the enzyme isoleucine. But when isoleucine is produced in a cell in considerable amount, the cell no longer requires isoleucine. Now this isoleucine, till it is used up by the cell, it will bind allosterically to this enzyme site. Means isoleucine will bind to the enzyme which is meant for binding threonine. So now threonine cannot actually bind to it. So in this way, exactly the cell is getting benefited. When you no longer are requiring isoleucine, why you will go on unnecessarily producing it? Because once you are producing something in a cell, a lot of energy or ATP, enzymes and many other things are getting utilized. So by feedback inhibition, the cells optimize the resources that they have in their hand. When you, simple fundamental thing behind this is that when you don't require something, then you don't produce it anymore till it is no longer required. So once it is more required, then this isoleucine concentration will be decreasing in the cell. Once isoleucine concentration decreases, it will not bind to this enzyme. Now it will follow up the enzyme and again threonine will attach to it. So feedback inhibition is a form of allosteric regulation where a metabolic pathway is stopped by the inhibitory binding of the end product to a particular enzyme which actually starts the pathway. So having said this, we have discussed actually 4 types of inhibition: reversible, irreversible. Irreversible is categorized into 2 types, competitive and non-competitive, and non-competitive type has a subtype to it that is called as allosteric inhibition and cooperative inhibition. And lastly, we have studied the feedback mechanism. So this brings us to the end of the series of discussions which we were doing on enzymes. Thank you.